Okay. Welcome everyone. We have Paul Cisek from the University of Montreal, and he's going to, I'm not going to do an intro or anything. We're going to launch straight into his fascinating paper on refining behavior with phylogeny.
Okay, thanks. So again, thanks for inviting me. I really so I guess what I'm going to do is I'm going to sort of do my spiel, but just stop me at places where you think something's interesting coming up. I think it'll be much more interesting if we do that and let it be a little bit driven more by your interests and specific questions than by the particular spiel that I can kind of spit out with one big breath. So one small explanation is a lot of this is material in a book that I'm writing. The book is quite far from finished, actually, and so I just sort of started publishing bits of it. So that paper is the first part. There's another one I'm submitting in about a month that focuses a little bit more actually on the kinds of stuff that Helen Barbas has done and a little bit more about mammalian and primate organization. And I can bring up some figures essentially from that paper if we want to discuss, because it's going to be a little bit related to the structural model and interpretations that might be a bit more familiar because I'll be talking about more recent animals. Anyway, so, and then there's all kinds of interesting dynamical systems thoughts to put in here, but that's not what I have in my talk. So if you want to have dynamical systems, stop me and ask me stuff or tell me stuff. Yeah, and we can do it that way.
And I put together some slides for the structural model. So if we have time, I can walk us through that, because
yeah, that would be great. Actually, I'm gonna sort of mention it, I think, when it starts to come up, but I'm not. Again, I might actually flip to the paper because it's actually has a lot more in common, and maybe some figures from the book that are just kind of thrown together. But anyway, all right, I'll start by sharing my screen. Here we go. You have my screen. So this is all right. So this is the phylogeny thing. Oh yeah. So I should maybe explain a little bit for those who haven't read the paper. There's a sort of a philosophical attitude that this expresses. I should outline it a little bit. And the attitude is that a lot of the terms we use in psychology and in neuroscience and in computational models of neuroscience are based on ideas about what the functional pieces are. So things like perception, attention, memory, etc. These are the sort of pieces in which we phrase the questions that we ask in neuroscience. But a lot of people, including myself, are becoming convinced that we don't have the pieces properly defined. And part of the reason is not surprising. It's, these things were defined by the ancient Greeks, by Descartes, by very brilliant people, but who didn't really know anything about biology. They were defined outside of biology, and they don't fit neuroscience. Now I could go through a bunch of examples on how they don't fit. But instead of doing that, I'm just going to go and describe another way of thinking about the pieces, and that is to ask, where do the pieces come from in an actual biological system? And obviously the answer is they come from evolution. They come from how evolution constructed the brain of a particular species over its long evolutionary history. And so the approach of the paper and the book and the talk is really to do that, to go through that history, sort of chronologically. So what this is here is the phylogenetic tree, a subset of the phylogenetic tree of animals expanded along the lineage that leads to humans. So you can see gigantic branches like all insects, which there's 930 million species here, are just collapsed into one. Fish, there's 40,000 different fish collapsed into one. The reason for that is because I'm interested in the lineage that leads to humans. You could do this for any lineage. You could construct such a tree for any lineage. And collapse other branches. But here what I'm doing is just showing how we diverged from different groups of animals over the last billion years or so. And it's not complete, but I'm sort of emphasizing some of the main transitions that there's actually a lot of knowledge about. So the branch points here are when we split off from different species. The thick bars are where there's fossil data suggesting it. So this is sort of a summary constructed from many, many studies, mostly based on genetic methods to compare species. And then what I'm going to do during the talk is just go through it. What happened, what were animals like here, what were like here, and what were the brains and nervous systems like? So that's what I'm going to do. I'm going to act, and it's going to be rather fast. And if you're not versed with all the jargon, it's going to be hard to assimilate. But the main point I would ask you to take note of is continuity and the progressive differentiation. In other words, you have a system which is simple, which has maybe one or two pieces that they gradually become more separated and specialized and subdivided and so on, constructing a more and more elaborate nervous system along the way as the system evolved. And I'll start way over to the left with the origins of life. So life began about 3.8 billion years ago with the first molecules. And there's a general consensus that what set life in motion was autocatalytic sets. So chemical reactions that have a kind of a closed loop where some enzyme catalyzes the production of another enzyme, which catalyzes the production of another one, and creating a closed loop. And that creates a kind of a metabolism and replication. So now the metabolism gets enclosed in a membrane. And you can imagine the metabolism really is like a negative feedback control system, right? So it just maintains the structure by producing the compounds as they vary, let's say, out of some kind of a set point. So you can think of this as a negative feedback system, and which it has to be in order to be stable and therefore survive. Now, you can imagine a couple of different types of metabolism. Once you enclose this system in a membrane, you can imagine that there's an internal metabolism, internal to the membrane. And that's going to exploit, evolution is going to discover that because it exploits consistent laws of chemistry. We would call that physiology. But you can also maintain your structure through processes that extend outside of the membrane. So suppose there's some, so here, whoops, excuse me. So here, this is some compound A that you want to keep in some desired state. But suppose there's some compound B and you don't have the internal chemistry to generate it or to control it. But what you can do, if you find yourself in a place where you don't have compound B, what you can do is simply move and absorb compound B from the environment, right? And this is just like physiology. This is a negative feedback control system, right? There is some condition that requires activity, and that activity then improves the condition. So I in the paper, I call that the impetus, which is kind of like a generalization of the concept of drive or stimulus, that any deviation from desired states motivates behavior, and then the behavior reduces or eliminates the conditions that motivate it. So it's negative feedback control. So behavior is, and this, many people have suggested this. Ashby, William Powers, Wiener said this in the 40s, etc. So the task of behavior really, you can think about it as complementing dynamics of the environment such that the entire system, organism plus environment, flows towards desirable states in the language of dynamical systems, right? You want to have something that sets up the dynamics to complement those of the environment such that the whole system tends towards a desirable state, like being fed, for example, and being safe. So we can think of all of behavior essentially in this way. Of course, it becomes more complicated. Okay, so anyway, so the first nervous systems appeared around here. They actually appeared also independently, it seems, in comb jellies. But for our lineage, they appeared around here, more than 700 million years ago, as a specialization of this outer layer of the animals that originally had these plurifunctional cells that responded to, let's say, tactile or chemical input through contractions, and then specialized gradually to have sensory, contractile, and these communication cells that essentially became neurons. So they would coordinate across the body the activity. And these animals had a diffuse neural net around them that was specialized into two particular subregions: an apical nervous system that was rich in chemical sensors, and a blastoporal nervous system around the blastopore that controlled contractions, either expelling or sucking in water. And these animals moved around, foraging essentially around the environment through something called Levee flights or Levee walks, where you sort of make small movements and every once in a while you make a large movement. This is a good way to cover non-uniformly distributed nutrients. And one proposal made by Thomas Hills is that this period of exploitation, that this shift between local exploitation and more long-range exploration was the original role of the dopamine, of neurotransmitter dopamine. So when things are good and you're absorbing nutrients quickly, dopamine is high and you sort of stay locally. But if dopamine drops, letting you know that you're not absorbing nutrients quickly enough, then you start making longer range movements. And this is, this he argues is true for pretty much all animals with neurons. And so the proposal is the apical nervous system is sort of sensing your state that you're in, and through dopamine controlling the blastoporal nervous system to make movements that move you locally versus more vigorously move you to a new site. Now, this organization can still be seen in cnidarians like jellyfish, anemones, etc. In our branch, the bilaterians, the body elongated, turning the blastoporal into a slit, which fused, creating a digestive tube, and the apical and blastoporal nervous systems merged at one end. Here's where the head and the brain.
I have a question. Excuse me.
Sure.
Yeah. Besides dopamine, when did the serotonin show up? That is, the animal needs to stop doing whatever it is doing. There has to be some satiation, right?
Yeah, that's actually probably even earlier. The serotonin system is probably even earlier. And as you say, it's more your sort of levels of satiation. So it's almost like your current nutrient state is signaled by, among other things, serotonin. And then your deviation from that state signaled by neuropeptide Y, which is also a very old neurotransmitter. And then those are sort of the higher level signaling molecules. And then dopamine is used to modulate your behavior in light of your state. Okay. In light of those things. So serotonin is probably even earlier than dopamine, before neurons, presumably.
In that case, norepinephrine and acetylcholine should be even earlier than these two?
I don't know. I don't think so. Norepinephrine, I think later, probably.
It's super complicated from what I've read, Rama. And there's also been a number of situations, as far as I'm aware, where precursors to particular neuromodulatory sort of systems have died out or kind of combined together, and there's all these really fascinating gene duplication events where, say, like the system for projecting a monoaminergic chemical out to the rest of the brain gets duplicated, and now you can use one of them for the original function, and you've got the other one set up to tweak just slightly to give rise to a novel function. And so even though dopamine or maybe octopamine came along before noradrenaline in a lot of the stories, the modern version of the dopaminergic system in the human brain and mammalian brains, where it projects almost exclusively to the striatum and surrounding tissues, as well as the sort of frontal cortex. The best story for that is that it came from a gene duplication of the A6 locus coeruleus system in I think some fish. I can pass all these papers around later if people can, but there's not really
there's actually a lot of
stuff
stuff about the various sources of dopamine in the hypothalamus as well. That like Mac is saying, that there was an original set of dopamine signaling that some of them remained or differentiated and others disappeared. Yeah, and it's a very complex story. I agree. Here's a little summary of what I was just saying.
We can follow up later. Thank you.
Yeah. Okay. Anyway, here's just a summary of this idea that the nutrient state is sort of signaled by serotonin. So the more serotonin you have, the less hungry you are. And then if you're hungry, then that's signaled by neuropeptide Y, which then in the presence of dopamine, which is saying like, I have food nearby and I'm absorbing the food, dopamine keeps you local, exploiting. But if dopamine is low, then you switch to this more exploratory behavior, right? And then notice everything is a feedback loop. So if you're eating, right, if you're exploiting, you're going to deplete the food, but you'll also improve your nutrient state. If you're exploring, you improve your chances of finding food. And so, essentially what's created here is this sort of nested negative feedback loops. And then
so I have a question about, I guess it's maybe naive and probably is explained by this diagram, but why is it necessary to have both 5-HT and neuropeptide Y when you can, I mean, they seem counterposed, but why do you need both of them?
I think they do have a different role. Neuropeptide Y is sort of telling you how far you are from your desired nutrient state, I think. It's a little bit more of a subtle thing saying hunger as opposed to just having food. Because you may want to, you don't always want to be completely full. Sometimes you need more energy than other times. And so you may want to dissociate this. I don't know, to tell you the truth, what the answer is. But I'm guessing it's something like that. It's like they're not exactly the same message. One is telling you a little bit what your nutrient state is, whereas the other is telling you how far that is from what you would like, sort of how far you are from the set point. But of course, there's many other signaling molecules involved in hunger. I'm just pointing out neuropeptide Y because it's a very common signaling molecule. And you can see, norepinephrine, one role at least of norepinephrine is to signal threat, for example, arousal, to say, don't bother feeding right now. There's more important stuff to do. So
Paul, can I ask?
Yeah.
I really like the idea of trying to kind of pull together multiple different neuromodulators and thinking about them as sort of almost working together rather than sort of in a potency. There's this kind of traditional story in the literature that dopamine codes for reward prediction error and that serotonin codes for blah, right? And to kind of give a single function to each of these chemicals. But if we remember that they've kind of come around in this sort of soup of general kind of pushes towards adaptive behavior or away from it, I'd be much more surprised if you could identify one particular function for one chemical versus the interaction between all of them providing a sort of
well, it's
framework.
the receptors that decide more. It's how you use the chemical that decides its function more, right? So there's different receptors for these things. But this does actually touch upon some of the reward prediction error stuff. I mean, here there's no reward prediction error. Here, this is a little bit more like tonic dopamine. It's the general level of how good things are. Are you in a rich environment or are you in a poor environment? It's really that kind of tonic dopamine level that this is talking about. The reward prediction error hasn't come into the game yet. It comes in, in a couple of 100 million years along this story. Mostly.
So my question, Paul, was, sorry, I got lost in a comment rather than a question, but my question was going to be, you also, if you're going to have these neurochemical systems around, you also kind of benefit, or an animal would benefit from keeping them under control. And so something like the habenula or the kind of epithalamus seems like a really important structure for that. Do we know kind of when it sort of emerged over the sort of phylogenetic tree?
Well, I know it's there in lamprey. Lamprey.
So it's a really old, yeah.
Yeah, it's very old. The habenula is very old. But I don't know if anyone has looked at its precursors in tunicates or Amphioxus. Those would be the next animals to look at.
That would be really.
I don't think anyone has looked at it yet, but it's possible that there are precursors. Although I don't know, because so much happens at the root of the vertebrates, and so many animals died out that there may be no precursors in tunicates. I'll come back to the phylogenetic slide.
Thanks, bud.
Where are we? Right here.
So,
So yeah, so we see it in lamprey, and so presumably, and it's very similar in terms of the circuit. So it's presumably around here, all these systems appeared. But notice this is a fairly long line. Lots of branches have died. The nearest branch, the tunicates, are very impoverished, neurally speaking, animals. They eat their own brain and become a giant stomach, right? So the next most useful animal is actually Cephalochordates, but that's like 180 million years here. So that's a big gap. A lot of innovation happens here, and we don't know that well where it came from. However, there are studies of Cephalochordates now that are suggesting some of the homologs that gave rise to things like even the Telecephalon. So there is a lot of interest, especially in the Amphioxus, to try to see the precursors of these things. So like a lot of the stuff on dopamine, for example. But anyway, I'm not going to go into that detail. I'll just move more quickly again. So the point is that you have this kind of these kinds of signaling molecules, primarily involving this apical nervous system, which became eventually the hypothalamus. So now this organization here is still seen in protostomes. So that's this whole branch that includes insects and mollusks. It's a gigantic branch, but I'm gonna go the other way to deuterostomes.
Can I ask a quick question?
Sure.
I feel like I was reading before that don't insects have an analog to dopamine? I think
yes, they do, octopamine.
Yeah,
and they do have homologs to a lot of these things because they've inherited. Whoops, they've inherited some of these old systems. In particular, the chemical signaling. Now, the problem, of course, is that so much has happened along this line that they've come up with lots of things on their own. And so it's very hard to really, there are homologs, and they're quite astounding in many ways. But it's difficult to really make conclusions because, they were doing that on their own. So for example, insects are pretty impressive. I mean, bees are impressive. Cephalopods, octopi are amazing. But at this stage, when we split off from them, they were nothing like that. We were nothing like that. They were nothing like that. This was very, very simple animals. So the potential was there, and they both made use of the same potential to do some of these things. But it's a different story, essentially.
Could I get a quick opinion, I guess? I saw a paper maybe a few years ago or something, maybe you've seen it, showing a homologous relationship between the basal ganglia and I think the arthropod, central complex or something. I'm not sure.
Yeah.
I want to hear your thoughts about that because I'm pretty naive.
Well, I have to tell you that in order to stay sane, I don't read everything about every animal because I would go insane. So I haven't actually looked into it because, again, my attitude is I want to know what happened along our lineage. And it's true that
homologs
tell us something what happened over here. But I sort of find I get a little bit more by staying a little closer to our lineage. The Drosophila has gone through a lot of niches, and so it has modified its nervous system in its way, in all kinds of complicated, interesting ways. But they don't have that much to do with what happened in our lineage, right? And so for that reason, I'm actually just not that informed about insects. I'm sure there's interesting insights, but again, I need to stay sane. Homologs with basal ganglia, I will let the basal ganglia specialists debate that with the insect people. I'll just listen to what they have to say. So I don't know yet really where I myself might stand on that. My guess is that the fundamental signaling molecules are probably very, very old. And so there is a real homology there, probably. but I think it's going to be hard to tease apart what's homology and what's convergence and what's just plain innovation that's independent. I can tell you one thing that at least my impression is from reading this kind of comparative work is that things are always older than we thought. So as science progresses, we learn that something is actually older than we thought it was. So it's quite likely, therefore, that we'll find out, yes, in fact, it's true. At least the functional organization of basal ganglia might be this old. But I'm gonna stay tuned on that. And in part because when you look at, for example, the Amphioxus, you don't see a lot of that there, right? And so, whatever happens over here, if it wasn't here, then it doesn't matter that it was over here. You see, so it's almost like a keeping the scope very, very specific to a particular lineage. But again, like I said, we tend to learn later that things are older than we thought. People thought the neocortex was a completely new thing. It turns out, you see homologs of it back here. So yeah, basal ganglia may be older. Certainly the signaling, the dopamine signaling. Although I do know, I can tell you one thing about dopamine is that I spoke with one of the people who's really the world's expert on dopamine evolution. And he says, one of the problems with dopamine is it's easy for it to be synthesized. And so it's very possible that it actually has multiple roles popping up all over the place. Mm-hmm. Anyway, yeah. So in my view, most of what we know about basal ganglia, what I think I know about basal ganglia, really only comes in here. Although some of the signaling, this general tonic arousal of tonic levels of dopamine seems to be much, much older. Maybe all animals with neurons. Although I think along the protostome branch, it's octopamine. It's a slightly different molecule. Anyway, in our branch, the body inverted and the nervous system folded into a tube. So once you get to chordates around here, they have an organization that looks a little bit like this. So it's like a segmented tube with a dorsal section called alar and a ventral section called basal. This is mostly sensory, this is mostly motor. And in fact, this is kind of like the dorsal horn and the ventral horn of the spinal cord, right? So the first segment or the first few segments of this tube result from the merging of this apical hypothalamic-like segment and the blastoporal. So these two systems, apical nervous system and blastoporal nervous system roughly correspond, the merging of them corresponds to the hypothalamus, and the rest corresponds to the rest of the spinal cord, the rest of the nervous system, which was mostly organized like the spinal cord, with a dorsal horn and a ventral horn. And there's these segments called neuromeres that then define sort of a rostrocaudal axis along the neural tube. And that's true for all vertebrates, all chordates. It just happens to be that some of these segments expand dramatically and others don't. And the whole thing twists and turns. But this is where Louis Puelles and Rudolf Neuenhuis define what they call the bowel plan, the basic plan of nervous systems. You can see that in the Amphioxus here. Which split off from us about 650 million years ago and appears not to have changed so much. This is why this is such a valuable animal, because it hasn't really changed in something like 500 million years, as far as we can tell from the fossil record. And it has many of the organizational features. So I'll just show some of those, mostly based on the work of Lacalle, but others as well. So there's a front segment called the archencephalon. It's a little bit like a hypothalamus. And the rest is called the deuterencephalon. This is like hindbrain and spinal cord. And dopamine centers are here in the hypothalamus, projecting mostly caudally downstream. And again, according to that proposal that I mentioned by Thomas Hills, governing this local exploitation versus long-range exploration type of movements. They also have, you can see in the Amphioxus, an escape circuit. So there's a single patch of photosensitive cells that projects bilaterally to this structure here, which has been suggested is homologous to the tectum or the superior colliculus in mammals. That then projects downstream. So whenever a shadow falls on this, the animal very quickly moves away. It has a sort of a separate set of descending commands for very fast movement, escape. So you can see that in the Amphioxus. You can see remains of it in tunicates of this circuit. Again, eye patch to the tectum. These animals are quite derived though. Then in the vertebrates, you really see things take off. In particular, one of the things that Ann Butler suggests was that one of the major things that happened is that the single eye patch split into two that migrated to the sides of the head. Now, if you have this kind of circuit, this escape circuit, and then you favor the contralateral projections, now you'll have a very effective escape circuit, because if a shadow falls on this eye, it's going to activate this tectum, which projects ipsilaterally downstream, so you'll turn away. So this is where Bratenberg's vehicles come into play, right? Now you're going to turn away and scurry away from the threat, likewise from the other side. And you see
a question here actually. So when the light sensitive groups split off to become the two eyes, they also stick around because there's a parietal eye in many.
So so it actually splits into three things.
Okay.
There's a pineal gland that moves dorsally to the surface, and the eyes go to the sides. In fact, the pineal gland you can already see in the Amphioxus. It's this thing called here called the lamellar body, and this is believed to be homologous to the pineal gland. So it's almost like the pineal split off first, and then you have this eye patch, and then the eye patch split into two that move to the sides of the head. And so that's kind of what like the parietal eye, and it'll stick around until we get to mammals. Anyway, and all of these parts are actually parts of the hypothalamus. This is still kind of the hypothalamus. But now you can start seeing in the Amphioxus, you can start seeing a distinction between, in this segment here, that's becoming a midbrain. There's a kind of a midbrain emerging called a dimesencephalon. It actually includes part of the thalamus, pretectum and tectum. And there's a border here called the zona limitans intrathalamica, which sort of splits the brain into these two segments. And that's still there in all vertebrates. Anyway, so now you have this escape circuit. Another thing, another thing that believed to have happened here is the expansion of the retina and the tectum, probably through gene duplication, actually, into a larger sort of retinotopic structure. So now you have some kind of a retinotopy, so that when you have light falling on one part, you turn more to the side versus less to the side, etc. But again, still ipsilateral projections downstream for the most part. In lamprey and other vertebrates, you can see a rostral part of the tectum as well, which projects contralaterally, right? So now if you have a contralateral input and contralateral output, you'll turn towards the source of stimulation. So now you have an approach circuit, which again, you can see this in lamprey quite clearly. And in lamprey, like many animals, the decision whether to approach or avoid something is made on very basic rules. Things like, if it's large and expanding rapidly, escape. In other words, the escape system has a high threshold, but it really dominates and shuts off the approach system. Whereas the approach system has a lower threshold, but it'll only act if the escape system isn't engaged. And so just by a simple thresholding process, you can make this decision between approach versus avoid. So you can see this in lamprey and in zebrafish. It's been studied very much in those animals. In fact, in zebrafish, the group of Herwig Bayer has really mapped the retinotopy of these descending systems for approach and escape very, very clearly. I'm only catching up with what they're doing there, but it's really nice work. Very, very much ripe for modeling, frankly. Anyway, so you can see that this organization in mammals, birds, you name it.
So regarding the escape behavior, one thing that I was not completely clear on was animals were still able to escape before this, right? So
Yeah, sure.
so the contralateral, so there's two crossings, right? There's first this the mapping of the input, the light input, and then this crossing. So you're saying that improved the ability.
Yeah, it improved it. So in the Amphioxus, where you have a single eye, you just run from light. Now there is some evidence that in the Amphioxus, there isn't really retinotopy in that front eye patch, but there is something like a motion detection in the tectum that perhaps is enough to tell it whether to go forward or backward. But it's not orienting away from the threat like a vertebrate does. The eye patch in the Amphioxus doesn't really have any retinotopy. Whereas here, even with a 2 pixel camera, you can just one here and one there contralaterally, you can escape very effectively. So it just makes escape a little bit better. And this was important because right here is where predation really took off. Until this point, life was pretty benign. But at this point, arthropods became predatory, and our own, vertebrate ancestors became predatory. So predation really took off here. And this is probably why there's so many branches missing, right? Because some of them were not so good as escaping. And there you go. But anyway, this circuitry has actually been retained, and you can see it in rodents, you can see it in fish, you can see it in I think every animal that's been studied. I think there's a very well conserved system. Always a contralateral projection, and then contralateral for approach and ipsilateral for escape. Anyway, now the other thing that happens in early vertebrates, because again, we can see it in lamprey, is expansion of the part of the hypothalamus here into the telencephalon. So this already in lamprey can be subdivided into an external pallium and an internal subpallium. So the subpallium is like the basal ganglia, a bit like the basal ganglia. The pallium is like every other part of the forebrain, including the cerebral cortex, but it's not differentiated yet. So the idea here, this is a proposal that I've made, but others have made as well, that here you sort of collect information, sensory, olfactory information, maybe visual information about your state. And then on the basis of that, you select downstream action systems like approach versus avoidance. And importantly, here's where you get this other role of dopamine, that you have a sort of source of dopamine that now becomes phasic and says, not just things are good or things are not so good, but says, what you just did was extra good, do it again in that context. And so reinforcement learning, which appears to have evolved around this time as well. Again, a big part of the reason why we made it through the Cambrian explosion. So the proposal is that dopamine now takes on the secondary role, superimposed over the tonic level of these little bursts that help to train this system here to learn when to approach and when to avoid and so on. But it's quite rudimentary. Now, already here, you can also subdivide this, though, into two segments. A ventrolateral segment, which so this is a proposal. Okay, the proposal is that there's two different, again, ancestrally already, there's two different ways to deal with your world. One is to stick around, and one is to go somewhere else. And so the proposal is that the ventrolateral part of pallium learned the cues for sticking around, all the things that you should be doing when you're locally moving around in your world, exploiting it. Whereas the medial pallium learned how to go somewhere else, navigate somewhere else. And again, it uses similar signals, but because it makes you move far, it learns where the good stuff is, where, for example, what odor plumes tell you where there might be a better site to seek food. Whereas the ventrolateral pallium is much more at the here and now, learning cues for approaching and avoiding things. And so this part here will become the hippocampus, and this part here will become much of the rest of the forebrain. Okay, then let's see. In jawed vertebrates, there's the cerebellum, actually, that comes in. And this is a major innovation happened around here, quite clearly happened around here. And one proposal by Montgomery and Bodznik and others is that it happened in a couple of stages. One of the problems of being a large mobile animal, as you're getting larger, at this point, our ancestors were still very tiny, but as you're moving more and more vigorously, the sensory consequences of your actions start to swamp your sensory input, and you become insensitive to things. One way of dealing with that is to cancel out the common mode. For engineers among you, the idea is that if you have lots of signals all varying together, maybe that's just due to you or something that you don't care about. You cancel that out so that you stay sensitive to the difference, the special thing, which might tell you where your prey is. Likewise, you can have the reafference principle, classic, idea of taking out the efference copy, subtracting it out, and therefore you subtract out the sensory consequences of your own actions. And then doing that in a more fancy way with some kind of a coincidence detector learning system, which now actually can predict sensory consequences of your motion. And this was important because if you're a large fish, as you're growing your body, you start to deal with more and more second order dynamics like momentum. And likewise, your vigorous swimming motions are going to swamp your sensory input. And so this circuitry allowed us to overcome those limitations and become bigger and become things like sharks. Anyway, so that was the adaptive filter model of the cerebellum. Very much like Mar, Albus, Grossberg type circuitry, predictive coding, very much along those philosophies. Prediction of sensory consequences allowed us to become bigger and faster. Now, I can get into the actual details of the circuitry if anybody's interested, because it's quite different in different branches. Then in bony fish, you get an expansion of the Telecephalon, and the parcellation of the thalamus. You now have two routes through the thalamus. The colothalamus, which is receiving information actually from the tectum. So this old structure for a visually guided locomotion projects through the thalamus to certain parts of the forebrain. But there's another part of the thalamus, the lemnothalamus, that projects directly, receives directly from the retina, projects primarily to the medial pallium for this, for navigation and things like that. So one proposal is that this information is just taking information from the tectum about the kinds of key stimuli that might stimulate approach behaviors or avoidance behaviors, sending them over here for learning, reinforcement learning, so that you can then later learn more effectively and through reinforcement learning, learn what to do. But you also have so there's some subdivisions here. There's a kind of an appetitive aversive axis. So the front part of this ventrolateral pallium. So the ventrolateral pallium sort of projects through the ventral striatum, ventral pallidum downstream, but also through caudal regions that actually will become the amygdala. So the amygdala is actually not a separate nucleus so much as the caudal end of the ventrolateral pallium. This diagram actually isn't quite right. I can show you a slightly updated. This will be relevant when we talk about the structural model. But anyway, the medial pallium, which will become the hippocampus, projects downstream through the lateral and medial septum for exploration behavior. But anyway, here you have this kind of appetitive aversive axis. So you're learning what's good to eat, learning what you should avoid and flee. Again, exploitation, exploration. Now, when tetrapods getting out on land, things change dramatically when you get out on land. Because you have one thing that's really dramatic is a much larger visual range. So you lose a lot of your ability to guide yourself by olfactory odor plumes. You lose the lateral line system, but you gain a huge visual range. And so now animals, but there's also many demands. Thirst becomes an issue. Body temperature becomes an issue. So many different things, but many different affordances as well that become very complicated, like finding a place to support yourself. Anyway, so there's a big expansion of the behavioral repertoire and a parcellation of the forebrain guiding that repertoire. So you have now additional subdivisions of the pallium. particularly the ventrolateral pallium, which includes the dorsolateral and ventral segment. And different branches did this differently. So amphibians, so if you take a slice through here, amphibians expanded primarily the medial pallium, because for them, navigation is critical. They need to stay near water, essentially. But they could use the visual information now, much richer visual information, to navigate with landmarks. And they didn't really elaborate these other sections so much. Sauropsids, which is this branch that leads to reptiles and birds, these animals remained diurnal. And this is an important difference between them and us. They stayed diurnal. They dominated the diurnal world. In fact, dinosaurs literally dominated. And so they expanded those tactile circuits, those same old tactile circuits that have been used since around here, for guiding visual motor activity, and the parts of the forebrain that received information from the tectum, so the ventral and the lateral pallium. So this becomes really large in reptiles and birds, called the dorsal ventricular ridge. And this is where you get a lot of auditory, visual, somatosensory information. Again, mostly, I think, for detecting the key stimuli so that you can properly modulate your downstream systems to approach and avoid the right things. But there's also a visual area in the dorsal pallium. The dorsal pallium will become interesting in mammals because that's going to become the cerebral cortex. But anyway, there's a visual area in reptiles and birds in the dorsal pallium. And I'm proposing that this part deals with affordances. And very simple idea. I'm assuming people know what affordances are, just the opportunities for action. And it doesn't have to be very sophisticated. And the example I'll give is this lizard here looking for a place to hide. He doesn't have to build a model of the world. He doesn't have to build an internal model of the world and then decide where a place in that model might be to hide. All he has to do is pick up on certain kinds of patterns in the visual input. So for example, if there is a place to hide, then there is a contrast edge between brighter and darker visual space. And as you move, that contrast edge will shift such that the texture above it will move more quickly than the texture below it, and the contrast edge should lie below the point of visual expansion. If these things are met, then it's likely that there is an overhang and that you can hide under it. And so my proposal would be that the dorsal pallium was doing these kinds of very simple rudimentary pickup of information that, not full visual imagery, but just enough to help you decide whether this is somewhere you want to run if you're afraid, if you feel threatened. So the idea of affordance maps in dorsal pallium. Okay, now the synapsids, which is the ancestors of marmals, these animals became nocturnal, in part because they couldn't compete with the dinosaurs. And so they became nocturnal, probably not until here, actually, maybe a little later, and warm-blooded. And now they reduced their vision. They lost trichromatic vision, and relied more heavily on olfaction, audition, and particular somatosensation. So they had hair on their bodies, first for insulation, but later for sensing. And that allowed them now to use somatosensory information to run around in the dark. And so they expanded the part of their brain that was receiving olfactory and somatosensory information, the dorsal pallium or the neocortex, which in mammals has an inside-out development that creates columns. And because of that, it can expand dramatically. And it did expand, of course, dramatically. Now, of course, the medial pallium becomes the hippocampus, the ventral pallium becomes piriform cortex and becomes purely olfactory in mammals. And the lateral pallium becomes insula and claustrum and some other structures, which we can discuss later with the structural model. Anyway, so here is a sort of a rudimentary map of early mammalian neocortex with this kind of central region that's granular and a ring of dysgranular cortex. And specialized for different modalities. So you have a lot of vision over here, somatosensation over here, audition over here. And it's organized into kind of a spatial sheet where information is spatially sensitive, processing is spatially sensitive, and a non-spatial sheet. So you can imagine that the spatial sheet can produce sort of action maps of the different kinds of behaviors that you can engage in, like run around, burrow for something, eat something, etc. Different kinds of activities. And the ventrolateral sheet specialized in key stimuli. So this, again, a little bit like what the ventral pallium was doing, detecting the stimuli in the world that motivate approach, avoidance, etc. Again,
I have a quick question here. It's kind of I guess a general question. It seems like that most of this, or maybe all of it, is divergent in the sense that I mean, it's all branching off from some ancient or more ancient system. Are there any examples of convergence where you're kind of melding two of the some of like the earlier regions?
You mean two systems that start to work together more? Well, in some sense, yes. I mean, there's the convergence of tectal and forebrain systems. So the holothalamus, that would be one example where you have this sort of ancient hypothalamic systems that just kind of saying things are good or things are not so good, and a midbrain system that's really dealing with visual motor control. But at some point, that midbrain system starts sending information over to that forebrain hypothalamic system to give it the information to have a better idea of what's good or what's bad. So that's one example.
The convergence is kind of in the connections then, rather than in the regions themselves.
Yes, that would be more in the connections, but of course the connections define the roles of the regions very much so.
Sure.
Another thing that's really dramatic, actually, and it's particularly true in this transition to mammals, is that the dorsal pallium in mammals, first of all, has much stronger projections downstream, but also it projects through the pallidum to part of the thalamus that then projects back to the dorsal pallium. This is the corticostriatal loop. And that doesn't exist in reptiles as far as I know. It does exist in birds, but it's probably independently evolved in birds. And so what happens here is now the forebrain system becomes, it's almost like you had this earlier basal ganglia system selecting tectal activities, like that those basic midbrain circuits for visually guided motion. But now that the connections through the pallidum back through thalamus and back to this dorsal pallium have become another system, a separate forebrain system that's now not so visual, but more somatosensory and an olfactory driven, but it's still doing the same role. It's still selecting between what thing to approach and sniff or burrow or whatever. So it's like it's taken on a new role by having this input, by getting this input. So it is connections, but the connections really change the role of the system. This, I think, is a very interesting topic that actually I'm still trying to get my head around exactly what happens in the mammalian transition, what the repercussions of that are. But it's clear that there's a much stronger interconnection with this closed loop in mammals. Now, the other example, I think, of what you're talking about is interactions between the hippocampus and the cortex, where this navigation system starts to become a more all-purpose. So at first, it's almost like navigation memory. It's cognitive maps of actual space that somehow becomes more relevant to all kinds of activities and all kinds of, much more high-level goals and things like that through integration with this part of the pallium. But I don't know that material very well yet. I haven't gotten to that stage so much. That's probably more relevant in primates than in early mammals. So that would be another example where things change dramatically because they become integrated and converge in a sense. And there's probably many more examples.
I have a question about this two cortical sheets, because this actually came up earlier in the lab meeting.
Yeah.
How do you decide that something is spatial versus non-spatial? Because in the paper you grouped some of the prefrontal areas with spatial, but there are not particularly good spatial maps in a DLPFC, for instance.
No, that's true. You know, that's kind of a work in progress.
Okay.
Maybe spatial is actually not the way to think about it. I think probably the better way of thinking about it is just the anatomical connections. There is a very different anatomical interconnected system here and a very different one here. This one's very closely integrated with the hypothalamus. This one is not. This one's very much primary sensory, visual, somatosensory, auditory. And this one, well, actually, auditory sort of straddles the two. This one's much more non-spatial senses, but also very much engaged with your internal state and things like that. So I think there's, you know, the prefrontal cortex, yes, that gets a little tricky, but there is a sense in which there's kind of a dorsal system and a ventral. But then where different prefrontal systems fit in, that's something that I'm actually trying to get my head around, and I'm hoping maybe you can help me actually, based on some of the connectivity patterns that Helen is looking at and others in her group, because it seems to me that ventrolateral prefrontal cortex might be over here, whereas the dorsolateral might be over here. Though, as you say, dorsolateral.
Yes, I
really
agree. So we can talk about that. I think that's right. What do you say?
I think there's something there. You know, I'm going to skip over to another slide just because we're talking about it. This is from this new paper. So we can sort of jump into this instead now. So this is kind of like the early mammalian state, based on the work of Kaz, Krubitzer, and others. You have a kind of a somatomotor region here and a visual region here, and it's surrounded by anterior cingulate, middle cingulate, and retrosplenial cortex on one side, and on the other side by this part here, which is agranular orbitofrontal, agranular insula, and perirhinal cortex. So this is sort of that sort of outer ring. Whoops. That's sort of that outer ring. And then this granular cortex would probably be somewhere over here, and again part of this system. So this is, anyway, this is a work in progress. But the idea is that the systems on this side, which are more related to ingesting and sort of internal state. So this is sort of the anatomical labels. Here's sort of suggested functional labels. That there's certain kinds of activity, like searching around for food, handling, burrowing, and actually eating the food. There's also at least some good reason to believe a defensive area. But then there's all this stuff having to do with your internal state and how it's satisfied by ingesting or escaping from something. The amygdala is kind of like the tail end of all that. And so then what you have in the early mammals becomes sort of balloons out so much that the whole thing gets curled around the insula over here. So the insula is over here, the amygdala winds up over there, all of piriform cortex gets sort of squeezed in there, and then you have this gigantic balloon of granular dorsal pallium neocortex, which then subdivides into many regions. But then I don't know exactly where does VLPFC fit in, where does this granular insula, I mean, there's a certain topology to this that anatomists are working out. And again, maybe this is something you and I can go
We can
through
definitely talk, and we can set up something with that.
But then if you look at it functionally speaking, what happens in primates then is they essentially expand this handling bit, right? All the different ways of dealing and interacting with your world, especially based on vision, just explodes into this huge map. So what was once, early mammals have these separate somatosensory map, but in primates, one of those maps just becomes gigantic spills into this expanding parietal cortex, premotor cortex, etc., for different kinds of activities. So I don't know if people are familiar with this, but there's a number of studies that have been done by Graziano and then later replicated in many, many other species by Kaz and Stepniewska and Leah Krubitzer and others, suggesting what they call action maps, so that there's certain patches of cerebral cortex dedicated to certain types of species typical activity. That has to do with the kind of things that monkeys do in their world, like bring things to the mouth, reach for them, walk around, jump around, defend themselves, etc. And this is very consistently seen in different species in kind of the same places. There's always a parietal map and a premotor map, and they're strongly interconnected with each other. But all of it is an expansion of, you know, what was here part of the sort of the early marsupial-like rodent behavior. Well, not rodent, but this kind of nocturnal marsupial animal that represents our ancestor. And then in prefrontal cortex is kind of like an expansion of these kinds of planning to higher level planning. But here things get very complicated. So here I haven't worked this out in any great depth. This is where Steve Wise and Passingham and Pruss and others are actually working out the idea. But again, if you think about it as affordances, then it makes pretty good sense. You know, as our repertoire expanded and the different affordances that we make use of expanded, so we expanded this part of our brain. Likewise, we expanded the temporal cortex now to be more than just detecting key stimuli like red berry is something you might want to eat, to actually do much more sophisticated visual object classification. But it's still really part of this kind of ventral lateral non-spatial system, right? This is kind of like the ventral stream, right? Of Milner and Goodale. It's taking information not to specify the spatial metrics of actions you might want to take, but rather take information to map your vision, to sort of identify the thing you're looking at and make, you know, an estimate of its value to you. Is this something you want to run away from? Is it something you want to approach and eat, etc. So that's yeah. Anyway, that's where we get to in primates.
I have a very good question. Oh, go. You can go first.
Sorry. Yeah. What you're talking about here in D, the center portion, would that be like in Penfield's map? That be like the sensory and motor homunculus? Yeah.
But the Penfield's map is not as clean as you know, sort of in a textbook. It's not simply body parts. There's a mixture. There's multiple hand regions, for example, and Graziano's proposal, and I think it makes a lot of sense, is it's because there's many different things you do with your hands, and you have actually dedicated in the primate dedicated system for those things. So there's a hand region for manipulating things and breaking nuts, etc., and there's a different hand region for bringing stuff to your mouth with your hand. And likewise, in the parietal cortex, there's separate sections. So there's a sort of it's sort of like a homunculus, but it's a bit functionally mixed up.
Or you can think of it as two parts, right? One for sensory motor processes and the other one for knowledge-based processes, where you're labeling the sensations into perceptions versus the other side taking thoughts into actions. Yeah, it's the knowledge domain versus sensory motor domain.
A little bit like that, except that these things are so strongly interconnected. And in fact, ancestrally, there were one thing. There actually wasn't, and there wasn't even a distinction between somatosensory and motor cortex in early mammals. That actually appears in placental animals only. So marsupials don't have it. And so this whole thing is kind of an integrated system that's sensory motor. And so there is, for example, lower thresholds for inducing movements by stimulating here than here, but you can induce movements by stimulating here. You just have to blast it a little bit more, right? And that's because it's all interconnected. It's a very reciprocally strongly interconnected network, of which part projects downstream. And so, some of these microstimulation studies, if you stimulate here, you get a defensive movement that's similar to if you stimulate here, et cetera. But there is a sense in which this is receiving a lot more information from prefrontal than this part is. And so for that reason, it is a little bit more, this is a bit more sort of like bottom up, and this is a bit more top down, if you want to think of it that way. There's a bit more influence here on goals and such.
Very interesting
Decisions
to try and
normally appear here before here. Sorry,
I was thinking about one of the few long-range projections that exists in the brain, goes from the prefrontal areas to the parietal, and this picture kind of helps you think about that a little bit, right? Because what we get with higher primates is a kind of this is going to be a little speculative, but where you have climbability, walkability, reachability, affordance is the right term. So you can just think about potential and how interoceptive planning coming up the other end of that particular dichotomy can now sort of exploit those potentials to sort of weakly evoke in a kind of memory sense, things that you might want to do. So planning, it would make a lot of sense to be able to incorporate things that aren't yet even in the environment. So when you're planning to go climb a tree that is not visible, it may help to actually sort of preload some of those affordances that you have on the right side of D. Does that make sense? Do you see what I'm trying to say?
Yeah, no, exactly. In fact, we were thinking with Giovanni Pizzullo, we sort of tried to extend this to a hierarchical level by saying, you have certain affordances that are available to you. Just they're there, they're in front of you, and you have a system that's very good at picking up on those. You've inherited from way back here somewhere, right? But there are also very, very highly predictable things, like, for example, a fruit that's out of reach, okay? There is a very, very predictable thing that any animal should be able to learn, and that is when you walk forward, things that were out of reach become within reach, right? And so you should be able to, for example, here, the proposal, of course, everybody would say, would be here somewhere. You here have a sub-goal, which is a predicted. It's like, if I want to make the thing reachable, what do I need to do? Well, I need to move towards it. And now I can make a long range, more abstract plan that is not related to things that are actually visible. It's related to something that I know about the world, something more like knowledge of the world and just the basics of, facts of life that, if there's a branch, and I know how to walk on the branch, I can walk towards the thing. So that's, that would be sort of an expansion of affordances into sort of potential affordances or predicted affordances or something like that. And then you can make higher level decisions. It's still only one step look ahead, but every step has to be a small one. So you want to have a primate that can handle reaching to things within reach. And then maybe every once in a while think about moving towards other things to put them within reach, et cetera, and expand this part with that. Now, it could be the cerebellum that's being used for that prediction. It's like a forward model of higher level activity, right? There's a forward model for what happens when you contract. When you contract your muscle, what's the feedback going to be like? This is a little bit like an outer loop version of that. If I move towards something, what's going to become available if I do that? Cracking open a nut makes the food inside available, etc. So that's kind of like you could at least, we think you could take that to that one step of planning, and then you have cognition, right? Well, simple cognition.
Charlie, you had something you wanted to say something.
QED. Yeah.
Yeah.
I mean, should I go?
Yeah.
Yeah. So First of all, this Graziano work is super cool, and it's very interesting how these patches are sort of oriented around a goal. I find that very fascinating, that you elicit goal-directed behavior by stimulating these. I wanted to just, one little thing I wanted to ask was, I wasn't aware of the work where you can stimulate these like higher order visual areas and you elicit these action maps indirectly. What's a good reference to?
Well, Krubitzer has done this very nice. Leah Krubitzer has done this. Omar Garbawi, although not so much on his own now. He did it when he was with Kaz. John Kaz he's sort of the mammalian evolution god, and Leah Krubitzer is the goddess, I would say. Although, I mean, there's no number of people, but they're sort of, and they've done a lot of this in primates. Leah has done this also in some other animals outside of primates. There's some work in rodents on this. Ivana Stepniewska works with Kaz. The two of them have done a lot of this stuff. And you can stimulate in parietal cortex. You just have to blast it a bit more. And so it's a little messier because you have to really, the thresholds for inducing movements are much higher here. And they've looked at, whether it involves actually primary motor cortex by cooling frontal cortex, for example, and doing things like that. So they're working out the actual circuitry.
Oh, that's a really interesting.
Yeah, it's very cool. We've done this a little bit in my lab. It's not that easy to invoke these movements, but some of them in premotor cortex, it's not so hard. And it's rather interesting because the animal doesn't seem to care. It's not like suddenly my arm is being controlled. It just does the thing and then goes about its day as if nothing happened, So it's
almost like it was its intention.
Exactly. It's like you can't help thinking that maybe the monkey's just thinking, yeah, I felt like scratching my stomach right now. That's why I did it. In fact, it'd be very interesting to do an experiment where we can ask that question. But anyway, so it's very controversial, mind you. But now I think after Graziano originally showed this and people were very skeptical, Kaz and Krubitzer and others, I think, have pretty much established that it's a real thing, and it's there in many primates, organized in a similar way. So galagos, for example, are primates that split off from us a very long time ago, and they have this organization. I don't have a phylogeny of galagos, but
have they tried this kind of thing in like rodents or?
Yes, in rodents a little bit. There's I think Brown and Teskey found some of it in rodents, although it's not so clean. It's not so clean. Rodents don't have much of a parietal cortex or premotor or anything, but you can evoke movements that seem sort of ethologically relevant. Anyway, so I guess that's actually mostly what I had. I mean, we've jumped past my primate slides. I can talk about some other things that if people want to.
Once you get to the cerebellum, there's no turning back, Paul. That's the place where you should finish.
That's true. I'm glad I put the cerebellum in there because I'd have a talking to from you, I'm sure. But anyway, so here's a kind of a summary of the of some of the major stages. Sort of my current estimate of where things happen. I mean, we can discuss some more on this if people are interested. But the basic idea again is that there's this. My goal is to have a progressive differentiation where the anatomy and the function are sort of hand in hand, constrained by where we think these things actually appeared, because the comparative anatomy is pretty solid. Even though it's difficult sometimes to compare between birds and mammals, there is a converging consensus about what the pieces are and how they evolved. And again, the Puelas work is central here, to me at least. So there is a certain logic to it. And then what I'm trying to do is essentially paint function on top of that anatomy in some kind of plausible way. And it's kind of in flux. but parts of it are very well defined. This part actually here is extremely well defined because there's so many good studies of lamprey. This part here is actually much trickier because not enough people are studying lizards. But then, of course, when we get to primates, then there's a lot of work. And so the goal is to connect it into a coherent thing that at least plausibly could be continuous. And I think then it tells us a little bit about the organization that's useful. So anyway, that's my evolution spiel, and it was only an hour and a half or something like that.
Well, we did ask a lot of questions, so thank you.
Well, yeah, I can pretend that it's your fault, but it was actually I took my time. Yeah, sorry about that. Anyway, I think I should probably let you take over, or unless people want to ask other stuff.
Yeah, I'm happy to do very quick because it's not so functional and I think conceptually not too tricky what I have to say. And I've mentioned it in passing a few times. So, yeah, if anyone shouldn't take too long, I can just quickly go through it. And it kind of starts off where you left off, so it fits in well because it's a sort of, in a way, orthogonal look at that map.
Mm-hmm.
Which is a sagittal map, so it's also literally orthogonal. But so Helen and the PI of my lab, Helen Barbas, has been developing these ideas for a while. That kind of started with her work with D. Pandya, and some of the progenitors of this were in the early 20th century. Friedrich Sanides kind of collected some of that stuff where it goes back to Dart and others. The basic idea, I like to introduce it in a way that's very like starting with someone who doesn't know anything about anatomy to say, so, well, what do you think about the cortex? What is it? And you get this Brodmann map, right? So Kobeny and Brodmann looked at Nissl stains, which I'll talk about in just a sec, and divided them up into these areas, these numbered areas that don't seem to have any particular connection to each other. The numbering is completely arbitrary. And so that's like one extreme way of thinking about the brain as just a quilt. And at the other extreme, you have what I think most people think, including a unfortunately large number of neuroscientists, which is that the cortex, the neocortex, is 6-layered. It's always 6-layered. And it starts with, I suppose, Mountcastle, but he was, right about the importance of functional columns in some parts. The place where he studied was cat somatosensory cortex, and there are functional columns linking the sort of vertical. And when I say vertical, I mean from the outer surface going in, because it's convoluted. It's a little bit hard to see sometimes. But later, even he started saying that, just the cortex in general is always organized in 6 layers. When you actually look at the cortex with any of the stains, you'd be hard pressed to always identify 6 layers. So Pascal Rakich, for instance, has been sort of fighting with some of the more exaggerated versions. Definitely, you can't blame Mountcastle for them, but people have been saying that the entire cortex is basically uniform, and our lab and the Rakich, a lot of people are saying, no, this is not the case. But it seems like people just want this to be true. Like if you look at someone like Jeff Hawkins, who talks about the brain, he's like the neocortex, and it's what you kind of want. There's a sense in which, if only it was simple, then we could make all these computational stories about functional Lego blocks. But even in places where the column has at least a reasonable amount of physiological data, people have questioned whether it's a helpful concept. And that's a kind of functional way to sort of start this story, which is that we really have to look at the specific circuits. We need a painstaking description of the circuits and the projections and response properties. And they're not the same because mainly because the internal connectivity is not the same. So when people say that the only difference between 2 cortical areas is that their external connectivity is different, that's not quite right. Definitely that's part of the story, but the internal connectivity changes because the cellular makeup also changes a fair amount. So how do people talk about this? The earliest methods were typically the Nissl stain. That's what Brodmann used. The nice thing about the Nissl stain is it only labels the cell body and nothing else, but it labels all of them. So you can do very nice counts of things like density, and you can see with the naked eye even the structure of the cortical band. So this is a sagittal plane. Sorry, so this is a coronal plane. And so you can see this ribbon of cortex. And what Helen and colleagues have done over the years is look at how the laminar structure changes. It changes smoothly as you go. And just by looking at the Nissl stain, you can see it. And what it looks like is this. Here, from this structural model paper, they picked a few areas in the, this is the insula, I think. Yeah. So the way that you can talk about it is in terms of certain features that are qualitative, but they also have a quantitative component. And part of what we've been trying to do is find good quantitative ways of talking about what the anatomists are pretty good at identifying subjectively, but with a lot of inter-experimental agreement. So the very simplest cortices have no layer 4. There's no granular layer 4 at all. So this is insula agranular. Can't imagine anybody thinks there's 6 layers here. Then you have a dysgranular, where you start to see it, what they call an incipient layer 4. Another thing you'll notice is that layer 2 and 3, hard to separate from each other. Layer 5 and 6 also not very easy to separate from each other. Layer 1 is always easy to see because there aren't any neurons in layer 1. Then you start to get a nice clear granular layer 4. They're granular because they look like little grains. They're these little round cells, not like the pyramids at all. And you can see this really well when you zoom in. So you can sort of arrange these according to what has been typically a somewhat subjective assessment, but the sharpness or clarity of the layers increases in a smooth way all along a band like this and everywhere in the cortex, basically. And so what we've been evolving in the lab since the 80s is types. And the types are sort of a discretization of what is more or less a smooth continuum. But this sort of simple concept, it takes a while to get clear. A lot of people think that if there's types, they're sharply distinguished from each other. But really, it's a pragmatic consideration that makes you divide things into types. So lately, we've been using between 4 and 6. I think 6 is more common nowadays in the lab. And this is a nice cartoon that shows you how the structure changes. It's basically what I just said. No layer 4 at all. And then as you proceed, the layer 4 starts to become pretty clear. 2 and 3 start to separate and become pretty clearly different. 5 and 6 also start to split. And then you start to get sublayers. So by the time you get to V1, you see you can separate 3A and 3B. Layer 4 starts to look like 3 layers. 5A and 5B can be split. So even saying that it stops at 6 is not quite right. So,
Johan, can I ask, does that, with the agranular and lesser extent the dysgranular, with the sort of very weak granular layers, does that mean that there aren't regions, cells there that receive what we would call kind of like core or sort of direct projections from the thalamus? Are they getting the sort of...
They'll receive a little, but they'll probably receive mostly matrix projections.
Yeah.
But those matrix projections will be to the bottom of 3, top of 5. They will still follow that. The core will still project in the way that you expect, more or less.
Okay. But there's just no like defined stellate-like cell population.
Right. Yeah.
Okay, cool.
Exactly. So it's not just, you don't have to just use Nissl. And now if over the years, people have found more and more markers, and in the past 10 years or so, it's kind of exploded. But another old one is myelin. So this is I think the same area again, the insula. And you can see that the extent to which the myelin penetrates from the deep white matter into the cortex changes quite a bit. This is a pretty reliable proxy for what they call the limbic to eulaminate spectrum. So limbic is kind of not that commonly used outside of our lab, but basically it means the agranular and disgranular cortices. And eulaminate means well laminated, so where you can definitely see a layer 4. And all these other markers are not perfectly aligned with the anatomist's subjective distinctions, but they kind of serve as proxies. And they, like myelin, for instance, it sort of works pretty well that low myelin means you're in a limbic area, but it's not always true. There are some, I think, yeah, there's one perial cortical area that has a whole lot of myelin in it. And another marker, which is on the right, is SMI32, which is a structural protein. I don't think anybody knows exactly what it does, but it's a one of those filament proteins, and it tends to mark mostly layer 5, the top of layer 5 and the bottom of layer 3, really crisply. And if you look at motor cortex, it's just awesome because you can see those huge bet cells outlined perfectly with SMI32. And this is a pretty good marker too. It works pretty well for going from the limbic, which has barely any of it. It peaks around V2. If you're looking at the visual system, for instance, it peaks around V2. But then there's a quirky thing happens where the SMI32 really drops off in V1. And we're still trying to understand this. So again, sort of a proxy for the sharpness of the layered boundaries. Now, one reason that we are interested in layers is not just to kind of categorize them, but also because they actually tell us a lot about how cortical areas interact with each other. And the first inkling of this was Deepak Pandya in his work with Kathy Rockland, who's still at BU. Deepak Pandya passed away just last year, actually. And he also was, mentoring Helen and a lot of this stuff. And so they found that if you look at how, say, V1 and V2 connect to each other, you see that there's a particular pattern where when you go from V1 to V2, that is 17, that's Brodmann area 17 to 18, you're going from the superficial layers mainly to 4, and maybe a little bit at the top of 5 and bottom of 3. And in the reverse direction, you're getting mostly 6 and 5 going to 1, 2, and a little bit comes from 2 also. And you see this sort of basically the same pattern showing up in the visual system as you go to higher up in the hierarchy. So I'm not going to go through all these figures, but it's basically the story is that Helen looked at this in the prefrontal cortex initially, but then started to expand this to study all the brain areas. Now, because of in V1 and V2, you can say easily which part is closer to the sensory periphery. So going from V1 to V2 is labeled feed forward, and V2 to V1 is labeled feedback, because V1 is closer to the LGN, so closer to the sensory periphery. And when Helen started looking at the prefrontal version of this story, she used that terminology. So I like to tell her, maybe you could call it the structural analogy model, because the real interesting thing here is it's an analogy, which is as V1 is to V2, so 46 is to 32. And unpacking that is the structural model, what that means and how it's a really interesting and helpful way to think about cortical-cortical communication. So when you have an area that's hardly laminated at all, that projects to a very laminated area, which is actually really rare, because the very laminated areas are kind of, Helen likes to call them like the snobs or the aristocrats. They don't often talk to people outside of their circle. You have a strong lopsided connection where the feedback goes from the deep to the very superficial areas, and the return connection, the feed forward connection, goes from 3, maybe a little 2, to the below layer 4.
Johan, can you repeat that quote? That was a great quote. V1 is to V2 as
as 46:32. Yes, V1 is to V2 as 46:32.
Okay.
You'll have the video anyway, so
Yeah. No,
I'll send that to you.
This one's worth writing down.
So
Johan, can I ask about whether or not Helen's looked into the specificity of the different cells and their feed forward, feedback projections, particularly the feedback projections, because layer 5 and 6 have got a bunch of different classes of cells. Some of them are only project within the telecephalon, other ones project out to thalamus and colliculus
about IT
and
versus
pons and
PT.
things. Yeah, exactly. Have they looked at that?
I don't think
Please
that
give me a clear answer, Johan.
we don't have not really, but because you have to do some intense double injection, triple labeling, it's not so easy to do that. But but what we do have is we're looking at the way that the corticocortical projections target interneurons. So that's part of the story. I didn't put that in this. I'll send you some stuff on that.
No.
But that's definitely part of the story. The PV neurons, the fast spiking interneurons, are more in the supragranular. They're present everywhere, but they're definitely more present in above layer 4. CB.
I have a question about the agranular versus eulaminate. Is there a evolutionarily phylogeny you can put into context for that? I'm coming to that because that's the more controversial part, but I'm going to end on that. So this is all very, very well established in terms of, so basically you can look at two areas. If they're connected, which you can't necessarily predict, if they are connected, you can say something about the qualitative nature of the prediction just by looking at the relative structure. So if they're not that different, meaning if you're moving to things that are kind of closer in terms of their difference of degree, then you'll see a more mixed pattern. So you will see the deep to superficial, but you'll catch a little bit of the layer 2, 3 also. So, which again, you can imagine as a kind of quantitative thing, which is the more similar they are, the more you're going to get a more mixed pattern of connectivity. And I have a mnemonic, which I've told Helen to use, but she hasn't used it yet, which is based on a balance metaphor. When you have a more laminated area, imagine that it's heavier. And in some sense it is because they tend to be more dense. But this is, again, a proxy. Cell density is not ideal because in motor cortex, which is a eulaminate area, they have very large cells. So if you look at the density, you get a kind of wrong number. So what am I trying to say here? That if you need to remember how the projection goes, remember that the one with the more layers is heavier, and the layers that see each other talk to each other. That's just the first level because the actual details are a little bit more complicated, but this just helps remember. So if you look at that blue area, it expands, meaning that when they are a little bit more similar to each other, you get a bit more of this crosstalk. And the feed forward and feedback projection. So the green would be feed forward, the red would be feedback. And here now, especially for this, it's a bit more of a caricature, meaning that actually there's a bit of mixing from 2 to 5, and maybe 2 to 2 and 3, and 6 to 2 and 3, 6 to the corresponding areas. Always remember that the cells in layer 4 don't project out. So the expansion, and then when you have areas that are roughly on the same level, that blue area expands, meaning that in principle, we can't really say whether it's an X pattern, meaning that the deep go to superficial and superficial go to deep, or if you get 2 and 3 going to 2 and 3, etc. But at some point you're talking about the same brain area, at which point you do in fact see local connectivity. So it fits even with that kind of perspective. So is this clear? Does this help?
Yeah, I like the metaphor.
Okay.
That's a very visually, visual mnemonic, very good visual.
Yeah, because I always found it difficult to remember which was which. And then it's like, oh, heck, maybe I can do this. So
could I try and reiterate it?
Yes, please.
I've just been a
lot here
for all these things. So you have the X pattern, And you're saying that if they're different, as in the leftmost
Yes.
picture, then it's sort of like the variance is sort of less as to where they go, but the X pattern is
Right.
still maintained. And then
Well, it's when I said X, I meant that the two areas are both feed forward and feedback with respect to each other. Meaning that, so with V1, it's communicating to V2
Mhm.
from it's talking to V2 from its upper layers. It's receiving what V2 says from V2's deep layers.
Yeah.
So that's what this captures here. So if this was V1, which is then these cells project mainly to 5 and 6. The return kind of corresponds when you look at it this way. Otherwise, it'll look tilted, like in this diagram. So here is the one where your laminate is heavier, way like the leftmost one that I showed. And this is the corresponding. So this is the feedback projection to upper layers, and this is the feed forward projection from the lower part of 3. And this is the more mixed. The idea is that you start to get exceptions to, so basically, if this is the most simple and exaggerated way of that very different cortical areas connect, this is more typical. You're gonna get a mix, but when you quantify it, you'll see that you get more of a bias towards the deep to superficial when you do the stereology and the counting, and you basically count the terminals and things like that. And that's what I'm trying to capture with the blue box here. The arrows don't actually tell you exactly where the layering goes. And at some point, it's a little hard to tell. Once you get to very similar areas, you can't know, because the injection will, for the dye that they use for these kinds of studies, will be taken up by the whole column. Very rarely can you get like just less than a column. Okay.
Johan, can I ask a question that's something that we've kind of pondered about in the lab, but haven't really had the time to like dig into literature to think about. So as you go from granular to agranular, the granular layers have got these really nice stellate cells that are receiving lots of core thalamic input, but then they starts to diminish across as you move to the agranular cortices. And when we ran an experiment in resting state fMRI study where we looked at genetic expression of just from a group of 6 people from the Allen Brain Atlas of different calcium binding proteins in the thalamus, right? The calbinin is supposed to be more enriched in the matrix cells and parvalbumin on the core cells. And when we looked at the correlations in the cortex to those patterns, it really recapitulated this, granular to agranular pattern really lovely. So, you can compare that to, T1 to T2 weighted imaging, which gives you a kind of indirect measure of myelin and a couple other measures. But the question is that something that sort of seems like it would make sense is that because the agranular regions are receiving less feedback projections, they're the ones doing the feeding back. That essentially what's happening over the kind of mantle is that you're at the one end or the granular end, the feed forward is coming from the thalamus, but the feedback is coming from the cortex. But then vice versa, up at the agranular end, the feed back is coming from the thalamus, but the feed forward is coming from the lower layer of cortex. And I don't know if that's an oversimplification, but it seemed to make a lot of sense based on our results. And as I said, I haven't had time to really dig in and think about it any further.
But yeah, just for fun, can anybody suggest an area that might be even more lower down the hierarchy than the granular cortical areas?
Well, it would be, what Puelas would probably call lateral pallium, and you would probably call allocortex or something like that, which is even, not really neocortex even, right?
Right. Or even hippocampus. And we've looked at the pattern for hippocampus.
Hippocampus would be another, yes.
So hippocampus to 25 actually fits the pattern to send feedback projections to 25. In fact,
that entorhinal cortex, Johan?
Entorhinal is pretty limbic, but I'm not
What's
sure
25 in non-Brodmann?
Sorry, 25 is subgenual ACC.
Okay.
It's below the genu.
So this would be, but this would be hippocampus, actual hippocampus proper, or like
CA1,
tecta, or one of those things.
CA1 and subgenual.
CA1. Okay, interesting.
Yeah, I can send you these papers. So it seems to work out nicely.
So you would place the hippocampus lower than the most granular cortices rather than, interesting. Okay, cool.
Yeah,
cool,
so Helen likes to think of hippocampus as like a 3-layer cortex.
3-layer, right?
Where, I mean, only 1 layer of pyramidal cells, but there are 3 layers. And in some sense, her speculation is that they sort of correspond to the deep layers. So again, this is more speculation, but the idea is that the deep layers are sort of older than the superficial layers. And the one really interesting thing that happens with the, I forget if it's all mammals, you get inside out development of the cortex. So you have, when the cortex is forming, you get these cells loading up in the subventricular zone. And then they initially form the deep layers. Then the next level, they go across the cells. So it's as if they're going past the older neurons in the cortex. So it's an inside out development. It's not like that in snakes and turtles. Layer 1 is sort of obligatory, is always sort of sitting there, and this inside out pattern is a relatively new thing.
Is that true for hippocampus though? It's not true for hippocampus.
I don't know whether it's true. I haven't talked to her about how
But I think that's a very much mammalian dorsal pallium specific, I think.
Right. And because and because it has only 1 layer of excitatory cells, I don't know exactly what to say about it, but yeah, I can ask her and we can talk about more about it.
That's actually an interesting question.
Isn't that kind of different than Phelan-evanescence story, where the hippocampus is sort of sitting at the end of the ventral sort of temporal pathway, right? You've got V1, V2, V4, TE, whatever. And as you progress along, then driving feed forward like inputs make it into the hippocampus, which then does a little weird pass off to the dentate, come back to CA3, CA1, and then feeds projections back down to the supragranular layers of the entorhinal cortex. So to me, like, that makes it sound like hippocampus is sitting on top of the visual hierarchy rather than underneath it.
Yeah. I
don't know. That's just how I was taught it. Yeah.
I think that because ultimately we're going to be talking about a closed loop of behavior.
Yeah, it doesn't really matter
where you place some of those things that are older is a matter of convention. So the functional hierarchy and this anatomical hierarchy, neither of them are true hierarchies, right? So you kind of want to be a little careful about having a,
Oh, sure. But replace the word top with then the hippocampus is receiving, quote unquote, feed forward driver inputs from supragranular layers of
entorhinal?
Entorhinal and from other kind of like higher ventral temporal cortices. So that wouldn't fit in my mind with the idea of the hippocampus as kind of in this balance metaphor sitting at the bottom of all of the balances. It would be receiving at the top end of a lot of the balances.
Well, okay, so maybe it's that CA1 is at the bottom with respect to those areas, and the dentate is a different story. So what I'm talking about is just CA1 and subiculum. So we'll have to, I mean, these are really good questions. So
sorry, I don't mean to derail the conversation.
I don't have a whole lot more to talk about, but these I will definitely try and dig up more on this and send it to you guys.
It seems like if there was going to be a place to have really weird connectional topology, somewhere where you're having to like navigate your position in the environment, and like whether or not where you are now is somewhere you've been before, is like a really great place to have a lot of these weird, strange loops as well. So it might just be that the hippocampus kind of breaks some of the rules because it's useful to do that, and animals that had that broken rule were more able to orient themselves in their environment, and then were more adapted. I mean, that could make sense to me as well.
Yeah, there's some stuff that I'm still trying to understand, both topologically and evolutionary, which I think I put something here about that. So maybe that'll help. So when you lay out all these types, they're not just arranged randomly on the cortical mantle, although because of the convolutions, it becomes hard to see. If you look at the medial surface, you see that there's a ring of allocortex, and then you have these agranular cortices, and you have this concentric ring structure. So most of the lateral surface will have pretty well-laminated cortices. And that was the macaque. Oops, sorry. That was the macaque. And now someone mentioned T1, T2. You did, right, Mac? Miguel and this was should be Zikopoulos here, but yeah, Bacillus Zikopoulos and Miguel Garcia Gapezas worked with an undergrad on this, actually a master's student, and also looked at the human brain using T1, T2, and they found that that's pretty decent proxy for for cortical type, and they were using, I think, the von Economo and Koskinas atlas to talk about type. They looked at the types. They actually scanned the von Economo book, did some analysis of that, and used that to talk about type. So that's pretty cool. So people are now collecting a lot of different data to do with the smooth nature of the continuum. And I'm also working on a manuscript right now to do with this, to try and come up with quick and dirty ways to assess the cortical type. And like I said, the cortical type is a discretization of a pretty smooth, usually pretty smooth. There are a few big jumps. Like V1 to V2 is actually a pretty big jump. An untrained anatomist will be able to actually see it. There's another one near 36, entorhinal, that area. So this is one of the most cited papers. It was the very first paper in the first issue of Cerebral Cortex, and it causes a great deal of irritation because Helen and some of her collaborators, Klaus Hilgetag, for instance, have been saying that this is underspecified, and actually if you use their criteria, you can generate hundreds of hierarchies that are not the same as this one. So beware of this figure, which most people, I think, put in their slides just because they want to say the brain is complicated. But using the structural model, they use the exact same data of element and Vanis and of what's connected to what, and they reorganized it according to cortical type. They divided it into 4 types. So this Death Star basically has the most limbic areas at the center and the most eulaminate areas on the outside. And the lines are showing the connectivity. So the black lines are connections between things that are the same area or one type apart. I think blue is skip2, and I think red is the long distance projections. So you see that as the structural model kind of shows, most of the projections are near along this continuum. You have a few long range, but when there are long range, the structural model will tell you what the cortical pattern ought to be. It's been pretty successful at that. And if you're going to pop something into your beginning of a talk, put this in. So now we come to this developmental and evolutionary story. Some of the earliest data that we've been trying to kind of sync with the structural model came from Pascal Rakic, and this is a review paper. He hasn't actually published this data separately, but they were looking at developmental maturation of cortical areas in the macaque. And so this one figure is all we have really, and very little, because it's hard to do this kind of thing in primates. So what this is showing how much of the cortex is sort of done, whether neurons have reached their destination. And we've got a somewhat limbic area, this one, and red, I believe, is V1. So red is 17, yep. So you're going from pretty not so eulaminated to the most eulaminated. V1 is the most well laminated structure. And what you see is that V1 takes the longest to be done. And this seems to be suggestive of something. The story that we're trying to say is that the eulaminate areas are the newest, but this is definitely not a consensus position. People will often say that V1 is homologous in all of the animals where you can name a V1, and based on so far preliminary studies, but from our perspective, V1 of a mouse is much more like V4 or dysgranular visual areas than it is V1. The most eulaminate area in the mouse is the mouse bowel cortex. And even that is not as sort of crisply defined as, say, auditory cortex in humans even.
So let me interject there.
Yeah.
So the other thing I think that's kind of the thalamic projections to these regions are quite consistent though across species. So like V1 gets this lemnothalamic, which would be the LGN, whereas V2 gets a lot of pulvinar, which would be like colothalamic. So this distinction that I mentioned, which is from Ann Butler, that very common across all vertebrates, at least jawed vertebrates. And it does seem to delineate a kind of a distinction between V1 and V2. You know, V1 is sort of like a relatively, it's relatively new in terms of like in mammals receiving direct lemnothalamic, whereas V2 is like a classic colothalamic, a little bit like the other parts of the pallium that also receive colothalamic. So the colothalamic is getting it from the tectum, not from the retina, getting visual input from the tectum and not the retina. Also auditory information goes like that. And that goes to those lateral portions of the pallium and the subpallium. Whereas the hippocampus and V1 get direct retina thalamus to cortex. So there's that seems to be actually relatively consistent, suggesting V1 is old, at least among mammals.
Right.
But then maybe became much more laminated in primates, no?
That's one, and I have been bringing this up with Helen a few times that if you choose to define the area by the thalamus, you might get the opposite kind of story. So I think what the response is.
Well, the idea that it became elaborate a lot more in primates is still consistent with that, right? I mean, it could, it's like a structure can be old, but then can be elaborated a lot more later and become really sort of newfangled, but
yeah. So
what they've been trying to say is that in the simpler, in the animals with less differentiated cortices, you have a lot more widespread connectivity. So so they're kind of inheriting a lot of connectivity and possibly losing some. So in the case of the V1 of the primate, it's much less connected to other areas than the V1. So that might be part of the story, which is that you're getting a lot more of a widespread projection from thalamus to visual areas. But also the thalamus scales with the cortex, and so does the striatum. Thalamus not as much, but and I was talking to Basil, and I just asked him briefly about this today, that you can also talk about how old different parts of the thalamus are, and not surprising, the core is newer. So that's than the matrix. So that also kind of fits with this idea.
Core is newer?
Yeah. I don't have a citation for that, so I can dig into. I can ask him for some more on that.
Yeah, at least in the LGN, right, the coniocellular cells are far more ancient than Parvo and Magno. So it's like Paul Martin, my old supervisor, always says it as like it's like a proto trial of like a cortical thing going on in the thalamus, the matrix. Yeah.
When I chatted to Ann Butler about this via email, the story is really, really complicated. It's like the neuromodulatory systems, right, where there's a lot of starts and stops, and then redevelopments, and then diminishings and all these different things. But it does sound like the matrix thalamic system that we see in distributed throughout the thalamus is a relatively mammalian invention, at least in our branch. I could pass those citations on. She makes me feel like I'm in kindergarten again with the amount that she knows about the thalamus. It's quite terrifying.
Yeah.
So that's what I thought, because I remember your exchange with her. She was saying the matrix is a mammalian thing, but if Johan is saying that core is more recent than that, then I'm confused, because I thought the core would have been there in I mean, from the beginning, from the beginning of the thalamus.
Well, the granular layers that the core targets were new. So, but I don't know a lot about this. So I'll bookmark it to dig into it more.
Yeah.
Um,
That's thrown a bit of a confusion my way.
yeah.
Also,
So,
speaking about V1, so V1 is so much larger in proportion in humans and I guess in primates than lower mammals. I'm just wondering how this all sort of fits with the speculation that V1 is sort of kind of a newer addition, so to speak. Or if there's any speculation about that.
Well, newer in terms of among mammals or introduced in mammals?
Or it's
because it's pretty much, I think it's pretty well accepted that it was there on all mammals. I mean, you can see something like it in monotremes, and it's organized sort of topologically, the way it represents space, very similar in marsupials as placentals. And so it's like, it seems that those people like Kaz and Krubitzer have concluded that it's there in all mammals. But then, of course, it gets very tricky when you go back further because there's such a big gap before we get to reptiles.
Did I misinterpret, Johan, what you were saying about receptive, or I guess receptive fields in like rodents in early visual areas looking more like V4 or something?
Well, they don't have a fovea.
They don't have a fovea, yeah.
Oh.
So you don't see the kind of magnification that happens with V1 in primates.
I see.
Is it possible in mammals, V1 expanded a lot more compared to pre-mammalian brain?
Well, the idea is that
newer, but expanded more.
The idea is that anatomically, it's so sharply defined that if you were just looking at it from an anatomical perspective, you wouldn't necessarily think that V1 of a rodent is a homologue of V1. But like you said, you could have development within something that is a homologue. So that's definitely possible. So this is not a lockdown thing, and we're not evolutionary neuroscientists, so we don't really necessarily have all of it down pat, but it's something that we're kind of sticking our necks out and suggesting that. And I think that the some of Puey's stuff is actually, they think it fits in with this overall story of the progressive nature of the origin of cortex. So the idea is that there's gradual emergence. This is something we haven't really talked about, but the idea it's sort of sitting in some of Paul's diagrams, that neocortex has this dual origin. There's one end of it, which is this ancestral olfactory cortex, and another end, which is an ancestral hippocampus. And the cortex kind of grew out of these two ends. And so the neocortical components that emerge from the dual origin show these parallel lamina gradations that expand in evolution through successive rings. And I'm going to show you those rings, which Paul also showed.
Yeah.
And so this D was what I was initially thoroughly confused by, because if you notice, the agranular is on the outside and the eulaminate is on the inside. So what this is a purely topological representation. It's flattened. So imagine that you've got like a rubber balloon that you're turning inside out, so that the mouth of the balloon is like stretched out to be on the outside. That's basically what's happening here. So with the agranular areas, you always find this continuity, so that an agranular area is not bordering any finely laminated area. So even though there's all these convolutions, this basic topology seems to hold. And new areas seem to show, new types of cortex, or let's say more refined cortices, seem to show up as islands inside the older, already present cortex. That's the guiding kind of thought here, which is why they make this sort of claim. I don't know whether they've specifically said V1 is the oldest, but that's sort of a conclusion that you can take from this.
That's very similar to what Puella says as well, right, Paul?
Yeah.
Yeah.
this is where, so they kind of, there was a synergy of this idea and what Perez is saying. So yeah, so it's this idea that you have this agranular sea of cortex.
We got a
and
slide for that.
it,
Yeah, nice.
within, yes, so yeah. The neocortex appeared in evolution as an island surrounded by allocortex. And the idea is that that particular idea just carries on, that a more elaborated cortical area shows up within, but only in this topological sense, because the volume of those things that are within are much larger. So you can think of it like a sort of really interesting explosion. In fact, that's what Paul was talking about with the wrapping around the insula thing, which is that it's like, imagine squeezing a cupcake or something. Stuff starts to kind of come out around, up and around. It's sort of what's happening. But in this cupcake, there's more stuff coming from the middle that ends up being the most voluminous. So
I think the mushroom metaphor that Helen used in that paper was a good one. You know, it's like you're looking at the stem of the mushroom, but then the whole thing just gets this huge head comes out of the center of it and just overwhelms and covers the rest of it.
Exactly. Yeah. So this idea which Paul kind of puts in his paper also, it's pretty controversial in biology generally. The idea of ontogeny recapitulating phylogeny. So you have, it's like a lot of people don't want to hear anything about it, but maybe there's a softer way of putting it is what I was thinking, because it is informing a lot of thought, and there's something to it that is worth looking into. So here over here you have the mammalian Telecephalon in development. These areas that Paul was talking about. Then you have the adult of I think a rat. This is a rat, and then you have a primate. And there's something that you could say that the in development what is it? A primate brain is like the end state in some sense of the rodent brain. That's at least it's a hypothesis to look into. So just as a last thing, I haven't actually read this carefully, but this is how Pueyes has been talking about this. So
yeah, this is a great paper. This is the 2019 paper from Pueyes. Yeah, it's very confusing, but yeah,
there's a lot here. So, but basically, it's this topological perspective that you have these more. So the as Paul was saying, this dorsal pallium kind of shows up as this border area, and then it differentiates further. So this is some of the markers that he was using to show the genetic markers to show this idea. It's the same kind of idea, and it's the same ring idea. But we're kind of taking that and just extending it within the cortex. That's the structural models evolutionary speculation, which is that the same idea just keeps going within cortex. That's the simplest way to put it.
So now there's a couple of things that I find confusing about the Pueyes model compared to Helen's model, and that is that Pueyes defines this thing called lateral pallium very distinctly, and that can be seen in lots of different animals, right? And then, but in this picture, the purple thing, right? The purple thing, which is kind of like agranular cortex in Helen's model, right? Yeah. But in Pueyes's model, he doesn't quite say it, but so the bottom part of it is what he calls lateral pallium. So that's where you have agranular insula, claustrum, and an agranular part of orbitofrontal. That part there, yeah. And perirhinal cortex.
Right.
In other work, at least, he suggested that's lateral, a different part of the pallium called lateral pallium.
Mm-hmm.
But then the upper part, which has retrosplenial, medial cingulate, and anterior cingulate, that's dorsal pallium, or is it, or not? I mean, it's, this is where maybe it's just a matter of labels, but it could mean a lot, because these structures have sort of, precursors. Like there was nothing like, was there something like that whole ring in amniotes? because he's very confident that lateral pallium, ventral pallium, dorsal pallium, and medial pallium are separate domains across all vertebrates. And now Grillner is showing it in lamprey as well, that there's a separate divisions in these four things. And the outer parts are ventral and medial being hippocampus, ventral being piriform in mammals. And in between them, there's these two other things called lateral and dorsal, of which the dorsal is the neocortex. But then my confusion is, okay, but that's not the same thing as Helen's model, because Helen's model is a ring that goes all the way around the neocortex. And so which part of it, I mean, maybe this is more of a question that I should ask Puelas.
Yeah, because this ring, I think the idea that Helen and Bacillus and Miguel were saying is that corresponds to the ring that they were talking about.
This is the agranular ring.
Yeah.
Right.
And you get just more and more of those as you proceed, which shoot outwards to the lateral surface. But
so you have that hem and anti-hem is the dual origins thing that's creating that neocortex island in the center, right?
Yeah, and I still have not fully understood how to think about the dual origin. I can't picture it. So I keep asking them, and eventually I'll get it. But yeah, I maybe if I read this,
there's a nice paper, a series of papers from Aboitis describing this. I forget what his first name is. Aboitis and Montiel have a lot of papers. I cite them in mine if you want to just look at Launching Point. And they have this BBS article about origins of neocortex with a lot of commentaries. That's a good sort of source to see what everybody thinks.
Is that related to Barbara Finlay's? Sorry.
No, I was
Go
just
ahead,
asking,
Sam.
please share the references. Thank you.
Oh,
it's in the paper, yeah. But Mac, you
Paul, is it related to Barbara Finlay's dual origin story? Is that,
I don't know. I don't think so, actually. Hers is, I think it's actually related because there is this ventral versus dorsal
that's right,
distinction that she makes, which almost sounds like ventral is kind of olfactory origins and dorsal is hippocampal origins. It's a little bit like that, it seems to me, and it also kind of connects to the fact that the ventral olfactory business is very much connected with the hypothalamus. So there was a paper from Murray and Averbeck, I think, Bruno Averbeck and Elizabeth Murray in Current Opinions or something. I can see what it is. They talk about sort of the two cortical systems very much, and it's a little bit like Milner and Goodale, actually. Again, there's a sort of a temporal, ventral, highly visceral and connected to the hypothalamus system, very much connected in with the basal ganglia, non-spatial, and topologically, again, kind of on that bottom limb of the Puelas diagram, towards the bottom limb of the Puelas diagram, versus a very much sensory-driven, external sensory-driven dorsal section that's things like, premotor and parietal cortex, spatial, again, a little bit like Finlay's spatial sheet. So I don't know, there seems to be a common theme that a lot of people are saying, this subdivision. But then, like we mentioned earlier, it's hard to say exactly where to draw the line, especially in some regions like prefrontal cortex. There is a very strong trend to say, and I think Murray and Averbeck suggest that dorsolateral prefrontal is part of the dorsal thing, and ventrolateral prefrontal is part of the ventral thing. They're very different. It's like that principal sulcus is a real big border between these two fundamentally different systems.
Right. That's what I was going to ask you. Do you know where to draw the border between these two ancient moieties? That's what.
It seems. I don't know. I think some people. I believe they would draw it at the in the prefrontal cortex at the principal sulcus. That ventrolateral prefrontal is part of that ventral connected system, together with orbitofrontal, ventromedial prefrontal, etc. Whereas dorsolateral prefrontal and premotor, and probably ventral premotor, are all part really of this dorsal thing that expanded so much in primates, and that's very spatial. And to me, it's almost like one is about affordances, one is about key stimuli, right? So one is for guiding movements and sort of specifying the metrics of it, and the other is more like decisions about what to do and what condition, like this is edible or it's not edible.
Paul, do you draw the link then with the distinction between declarative and procedural memory as well?
Well, I'm not sure I would use particularly that distinction. I think that's a troublesome one, but a little bit, yeah. There is a little bit like there's a system for performance and sensory motor control that's more dorsal, and that's all frontoparietal cortex, those action maps and all that. But then there's this other thing which is much more ventral and much more about detecting objects in the world. It's more about identity of objects in the world.
It's the where versus what? Where stream versus what stream?
Yeah, it is actually. It's like the what versus where stream is kind of the visual caudal end of a larger distinction in cortex, which is a sort of a dorsomedial ventrolateral distinction.
I think Grasberg uses that, right? Yeah.
Yeah. No, I think it's I mean, certainly that distinction. Yeah. And
I always prefer
distinction is
calling it the how pathway rather than the what is it, the where?
Within the where,
like
yeah.
how
No,
you
I'd rather
interact,
call it,
how to interact with it, like an
I'd
affordance.
rather call it how, yeah. Because it's very much about sensory motor control. Yeah, but then where they converge in the frontal cortex is tricky, which is the frontal pole part of one or the other. I mean, there's a lot of anatomy there to be done.
Could that be the end of the road? Where everything meets,
pole?
both streams meet. Yeah.
I don't know. Actually, I think the frontal pole is probably older. I mean, that's a.
No, I'm saying that's where the streams converge and collective understanding happens.
Yeah, I don't know though, because the one study that recorded in the frontal pole in monkeys concluded that it's actually pretty boring. The frontal pole is just signaling what you just did, essentially. They said the frontal pole, you'd think it's the top of the hierarchy, and so that must be like the seat of consciousness or something. But no, it's actually pretty boring.
There's some speculative stuff that I noticed about how the visual and auditory areas start to come together if you follow the trajectory into the prefrontal cortex. And one way of thinking about it is that, which is wildly speculative, is that you, just by virtue of kind of anatomical proximity, you may create the following sort of affordance, which is that you get the best of visual object recognition and auditory motor sequence planning, and that is language in some sense. So you take your fine object recognition system and you bolt it onto your sequence generation system, and you get at least the linear output side of language. But I mean, that's why I have no idea how you would ever study something like that. But it's nice to think about how multimodal systems become amodal.
I think
actually that's something that I thought about as well. And the interesting thing is that if you look at the comparative data, a lot of these systems were actually multimodal before they were unimodal. So like in amphibians and such, the thalamus doesn't really have modality specificity. It's just this big thing that projects everything. And in some ways, maybe that makes sense, right? Because if you want to learn about what to do, you want to take every single bit of information you can. And modality specificity is really not that important until you want to do more than that, which is a little bit more fancy pre-processing along the way. So you have some more invariances that are purely visual. You know, there's certain things about vision that don't apply to audition. And so you kind of want to, it's almost like you want to start off with something which is multimodal and then gradually separate the front end to become more unimodal so that it can process things like, you know, size invariance or something for vision differently than frequency invariance for audition or something, right? But fundamentally, the older thing is the multimodal thing.
Exactly.
I wonder how
maybe that makes sense.
I was wondering what the perspective would be from fMRI, where they have the intrinsic networks shown by functional connectivity, which suggests that the default mode network is maybe the most multimodal, and it's, you know, distributed. It's got the subgenual anterior cingulate and the PCC and you know, lateral temporal lobes. It's got these distinct hubs, as well as all the other areas that doesn't seem to really fit in well with Helen's structural model at all. They're just completely separate ways of chopping up the cortex. And also in terms of evolutionary phylogeny, the intrinsic networks from fMRI seem totally separate. And so I'm wondering how to reconcile those completely different atlases of the whole brain, really.
Yeah, that's a tough one. I mean, I think the one of the difficulties is though that most of the studies, I mean, the default mode network is a bit more unstructured. You know, just look at activity and see what correlates with what. But a lot of the studies in MRI are trying to tap into the kinds of tasks that might actually be, you know, almost impossible to understand until we actually have a lot more knowledge about the brain, right? So the strategies with which people do the things we do are just going to be weird. But the default mode network, yeah, I mean, that's an interesting thing because it does seem to involve a lot of the outer ring of Helen's model, topologically speaking, outer ring, right? You know,
but not all of it.
Well, no, but insula, the usual suspects, ACC, insula, RSC, you know, they're always light up in these systems, and that's kind of that outer purple ring, right? That limbic ring, I guess you could call it.
Yeah, the ACC and insula are sometimes kind of grouped together, Paul, with other parts of lateral parietal cortex, which I don't think is
lateral parietal would not.
Laminate. Yeah,
no, that's true.
And then parts of the default mode will have that kind of a granular feel, the retrosplenial cortex and parts of the subgenual anterior cingulate. But then again, there's other non-agranular regions like the superior mesial frontal cortex, other parts of the parietal operculum as well. So I think I kind of feel for you, Kale. Like I think linking these things up is going to be really challenging. And I think probably one thing to remember, right, is that what Helen and Bass and others have been sort of arguing for with this structural model is sort of like the rules that govern the interaction in terms of connectivity, but they don't necessarily tell you about how you've ended up using your brain over the course of your development. And it might just be that the default mode, because of this weird analysis we do, shows you something about how you use your brain in general, which is something like have evaluation of some memory and, you know, think about elaborating upon it. And then you just sort of end up getting Hebbian plasticity between them. And it's not because the system had some, you know, privileged flow of information. It's because you used it in a particular way. And so it's very, very hard to disentangle these things, right? They're just, we're analyzing, you know, people unconstrained in a scanner and try to like make these really strong kind of claims about it. But it's just really, really hard to do that at this level of granularity that we're looking at with the structure.
Yeah. I think that I don't want to keep anyone since it's 11:30. So Paul, if you need to run,
it's only 1:30 in Australia. We can keep going for hours yet.
Yeah, I'm a night bird, so. But I think that I don't know that much about the parietal cortex. I haven't really looked into it as much, but the fact that you have these long range connections to it and that it has such a close relationship to the prefrontal cortex is telling us something, right? And if you're saying that we had a few of these agranular areas or disgranular areas and this one reasonably eulaminate area,
then you kind of want to ask about a slice through this cake, right? Which is like, how do you make teams out of this? Like, it doesn't make sense to think of all the laminate areas as doing the same thing.
Oh yeah, no, I don't think so. I think you're right. Yeah, I think you're right. I mean, it's like the parcellation of function is going to follow along all kinds of little idiosyncratic paths as this thing expands. Yeah.
So
are you familiar with the memory book by Steve Wise and Betsy Murray and Kim Graham? So they have a nice concentric ring development there that I think is very much in line with Puelles, but also with the structural model. And they discuss, and the Passingham and Wise 2012 book has this too. So this book is Murray, Wise, and Graham, 2017, I think, The Evolution of Memory Systems. It's a great book. And anyway, but they very much are starting off with this concentric ring idea and the addition of the internal regions, right? So you have this sort of limbic ring, and then this granular ring, and then you have this sort of what Helen calls eulaminate, and it. But then within it, different islands appear at different times. So like caudal prefrontal appears before dorsolateral prefrontal, and there's a subdivision that occurs there. I sort of hinted at it when that slide I flashed up, but they developed that in quite some depth. Again, by looking at a number of primate species. So where those prefrontal regions appear, they work out in some depth. But I guess, again, I'd like to see anatomical and developmental studies showing this, especially in terms of sort of large scale connectivity. So Bruno Averbeck is doing some of this stuff. Keep an eye on his work. He's looking very much at the large anatomical systems in macaques. Be nice to see. You go ahead.
Sorry, when thinking about the function, I have a sort of anthropomorphic story I like to tell about this it's progressive division of labor, and if it's true that what we're saying is happening in development is also happening in evolution, then it's sort of like this, that your ancestral or the most limbic prefrontal areas, which are very close to motor areas, are like aristocrats 1,000 years ago, in the sense that the aristocracy's always been around, but what they do is changed over time. So they were more jack of all trades earlier, but now with the addition of these new areas, it's sort of like the creation of factories. So now the things that the managerial parts of the brain get are already heavily pre-processed. Mm-hmm. So the way that they are able to make their mappings between, so the key stimuli are more refined, and the control over the individual parts of the effector system are more refined. So what the intermediate, the ancient areas are actually doing has altered as a result of not needing to be micromanagers of the entire body. Mm-hmm. And that's my sort of anthropomorphic way of thinking about it.
Yeah, no, that's actually not, that's a cool way of thinking about it in terms of,
and that may actually also provide a way of thinking about new areas being sort of given the right to take over certain anatomical connections. So it just struck me that if we're trying to say that V1 is a sprouting off of V2, then presumably any part of the thalamus that was visual was in contact with V2 primarily, and then V1 just inherited those same anatomical structures. And then you could imagine a situation of pruning, but on a slower time scale, of evolutionary time scale, where V1 sort of proves itself and takes over all of the LTN connections. Mm-hmm. That's another thing you can think about, that inheritance plus sort of the restriction and taking over.
Yeah, it'd be good to do that again comparatively across like monotremes. Anyway, this has been really interesting, but I do need to
It's
go,
late, yeah.
and I guess everybody needs to
Yeah,
go.
we all need to go. Thanks so much. This was awesome.
Yeah, thank you.
Yeah, this was
a great
amazing.
Yeah, thanks, Paul.
I
That was
enjoyed the
awesome.
discussion.
Yeah.
I'll
And
send you
maybe
the
I'll pick your
video.
brain a little bit more about what else, because some of the I took a lot of screen snips of your talk.
I'm gonna send you the video anyway, and I can send you the presentation.
Yeah, that'd be great. Yeah, that'd be great. I'm
So,
gonna look up the papers you mentioned. as
and
well.
I've written down the stuff I need to ask Helen and Bacillus, and we'll solve this V1 issue. Okay. All right.
All right, cool.
Good night,
Anyway, so
everyone.
Thank
so
you.
thanks again for inviting me.
Paul,
Thanks,
Johan.
Max, for